Uh, Good morning, everyone. My name is Yuyang Qiao, and I come from the Shanghai Jiaotong University, Shanghai, China. And today, I'd like to introduce a new software designed for the metagenomics sequence classification, MetaBinG2, especially deal with the samples with many unknown organisms. And this is outline of my presentation. Firstly, I'd like to introduce the background of metagenomics sequence classification, and then I will introduce the method and algorithms of MetaBinG2 and the dataset we select to test it. And the third part is the result part. I use this part to show the performance of MetaBinG2. And finally, I will discuss the extension of MetaBinG2 and the future application of it. Okay. As we all know, the microorganisms are everywhere in the world and occupy the very important position in the ecosystems. But however, we can only capture the little knowledge from them because the small fraction of them are countable. But when we face the uncountable genomes, what we should do, luckily with the development of metagenomics, scientists can collect the DNA directly from the environment, and sequence technology brings us a huge opportunity to find the hidden world behind the diversity of the microbial world. And when we obtain such huge size of the sequenced data, the first question we want to know is what is in this data and what are their proportion. And sequence classification is aimed to deal these questions, and is first and one of the most important question in the step in the metagenomics analysis. And for sequence classification, there are several softwares have been developed to do these questions, and they can be divided in mainly two parts: the alignment-based method and computation-based method. Also, some researchers combine these two strategies to develop their hybrid method, but this is not the point of this presentation. And I will make the brief introduction about the alignment-based method and computation-based method. Alignment-based method uses sequence homology, align the short sequence to the reference, and if the source genomes of the short reads are included in the database, the accuracy of alignment-based method are higher than the computation-based method. But when their source genomes are not included in the reference database, this accuracy of alignment-based method will drop dramatically, and this conclusion has been demonstrated in the previous work in FIMBL. And besides this, expect for the current method like the Kraken and clock based on the K-mesh search algorithms. Most alignment-based methods, especially the BLAST-based method, are too slow to classify the huge size of the sequenced data. And for computation-based method, they use sequence patterns like the K-mesh frequency to find the regularity between of the sequence and use this regularity to classify the reads. So they don't have such high dependence on the known genomes. And compared with the most alignment-based method, especially the BLAST-based method, they have advantages on speed. No matter what kind of strategy we choose, we have to face the two challenges. The first is with the growing size of the reference database and the increasing size of reads generated by the modern sequence technology, we need more computing results, memory, and disk space to solve these problems. And all this make the speed be a non-negligible challenge. And besides this, although the size of the reference database is continually increasing, the known genomes still only occupy the small fraction of the microbial's world. So how to do the sequence classification for samples with large part of unknown genomes within the limited time is our destination. And to deal with these questions, for classifying the unknown genomes, we choose a combination-based method, and for the speed part, we want to input the GPU to accelerate. And based on this, we designed MetaBinG2. And this is the algorithms of MetaBinG2. Firstly, we converted a complete genome sequence into a state transition vectors under the case order Markov model. And a state in this Markov model can be defined as a sequence with length k, and each state can transfer to 4 kinds of other states. So there are 4 to k plus 1's power kinds of transition probabilities. And the transition probability is transferred from the state M to state N of the genome I can be calculated as this formula. And OM and ON represent the oligonucleotide of length K, and the PR represent this transition probability transferred from the state M to state N. And after this conversion, we can calculate the distance between the short sequence and the genomes. Here, I should mention that the previous work has demonstrated that most softwares designed for the sequence classification have the character that they often over-predicted the result. So it caused the false positive relatively high. To solve these problems, we wish to input the samples information. So we make the new assumption that a sequence is more likely from the genomes, which take a large proportion when the distance between this sequence and several genomes are familiar. So based on this, the distance between the short sequence with length L and the genome I can be reflected by this distance score Si. And here, omega I represents the weight of the genome I. Then the converted matrix of the short sequence and the genomes will be used to do the matrix multiplication and get this score matrix. This is the systems of MetaBinG2. Firstly, we transferred the initial genome sequence under the Markov model, and when we run the MetaBinG2, the loaded database and the converted vectors of the sequenced data will be uploaded to the GPU memory and do the matrix multiplication and get a score matrix. And here, each genome's weight will be used to adjust this score matrix, and when each genome's weight tend to be balanced, the optimal scores will be annotated with their taxonomy level and output. In order to show the performance of MetaBinG2, we designed this benchmarking test, use the Clade exclusion experiment, and I will explain what experiment is later. And here, we prepared 26 and 6 microbial genomes to build a database and selected these 4 softwares to do this benchmarking test. I should mention that all these 4 softwares we selected have advantages on speed. For clock, it's one of the alignment-based method based on the k-mesh search, and the MetaSUB is a composition-based method. MetaBinG is the first version of MetaBinG2 and was published in 2011. And at that time, it's 1500-fold faster than another famous computation-based method, Phi. And we use accuracy to measure each software's performance on this benchmarking test. Okay, I'd like to use these figures to explain what a Clade experiment is. This experiment is designed to evaluate the software ability to classify the unknown parts of the samples. Here, we named this database no excluded database. It means the genomes appeared in the test dataset are all included in this database, and we have no unknown part in the test dataset. And how to design the Clade excluded database? For example, when the genomes of in the test dataset belongs to the order A and order B, when we build the order excluded database, it means we removed all the genomes of order A and order B from the no excluded database, and use and through these ways to simulate the unknown part in the dataset. Okay, next I will introduce our datasets, the 4 datasets. The first one is a simulated dataset, and the community distribution of this dataset is coming from the published paper, and in the previous work is designed to evaluate the software's potential to classify the samples with many unknown organisms. And we use the NASM developed by our lab to re-simulate this dataset with 29 gigabytes. And as we can see, it's such high size. It's a huge size, and this dataset is the most uniform distribution. And the second dataset we selected is among data. It means, although we don't know the exact source genomes of each read, but we know the distribution of this dataset because scientists mixed several species with a predefined distributions in this dataset, and we will use this dataset to measure the performance of 4 softwares later. And the 3 dataset is from the real-world sequenced data. It's sampled from the core Roman. And in previous work, scientists used the assembling method and Bing method to identify the unknown part to forming phylogenetic orders. And I listed their information here. It's very useful to compare with the result of other softwares later. And the 4th dataset is provided by our conference MetaSUB dataset, sampled from the three cities subway different position. Okay, next I will introduce the result part. I divided in this part in this result in 3 parts. The first one is the result of benchmarking test, and then the result of correlance analysis. The last one is analysis of MetaSUB dataset. Okay, this is the result in the Clade exclusion experiment. When the genomes in the test dataset are all included in the reference database, we can see clock has the best performance. And the second point, we can see that compared with the first version, MetaBinG2 really improved a lot on the accuracy, and the read length is benefit to all these 4 softwares. Sorry. This one, I'm so sorry. This is the result when we use a no excluded database, and the clock is the performance of clock is the best on the accuracy, and MetaBinG2 has improved than the first version. And when there is some unknown part in the dataset, MetaBinG's performance better than the others. And we use these figures to show the all excluded level of Clade exclusion to show the performance of these 4 softwares to evaluate their potential to classify the samples with many unknown parts, and we can see the MetaBinG2's performance is the best. We also calculate the consistency between the predicted distribution and the truth on the simulated data and mock dataset. From this direction, we can see the distribution predicted by the MetaBinG is the best one, and from the false positive, the MetaBinG2's performance is also very good. For another part, the speed part. For the fair comparison, we applied all these 4 softwares on a same computing node with a CPU with 24 cores and 1 GPU called Tesla K80, and measured their performance with on the second level and dividing in 2 parts: time for load database and time for classifying the sequence. And we can see all these softwares can classify such size of simulated data within 3 hours. It's so fast. All these are so fast. And here we can see Clade has amazing speed. And later I will discuss its performance on the real dataset to see its performance later. Here we can see this is a result of these 3 softwares performance when they applied on the real world sequence data. And although Clade has amazing speed, but there is a large part of its result unclassified. We think it influenced us to understand the outline of a sample. And for MetaBinG2, here I mentioned this reference phylum is identified by the previous work, and the part covered by the red circles means this result of the softwares is same with the reference phylum, and we can see the MetaBinG2's result is a close one to the reference phylum. And from the class, the tendency is the same. MetaBinG2 is also the best one. And from the order set, we can see the conclusion demonstrated by the previous work, MetaSUB and the Clade over-predicted. They have much more orders. Different with identified orders before. Okay, this is summary of result 1 and result 2. From these 2 parts, we can see MetaBinG2 really have the advantages to provide the outline of a sample which has many unknown organisms, and the speed of MetaBinG2 is also very excellent. The last part of the result is the analysis of the MetaSUB. To analyze this dataset with such high complexity, we add some eukaryotic genomes to our database of MetaBinG2. And the first point we are focused on is the mean phyla distribution of each city. The first, we selected the phyla whose average percent are more than 1%. And for each selected phylum, we count the number of samples and all this phylum and calculate this ratio. And phylum appeared in more than 50% samples will divide as the main phyla of the city. And from this result, we can see New York's complexity on phylum is low. The main phyla just be the justice, the proteobacterial, and for Boston, the main phyla is actin bacterial and proteobacterial. And the main phyla of Sacramento are more than the other two cities. They have such these phyla included the eukaryotic genomes like the chordata and the streptophyta. The main phyla are very different among these three cities. The second point we focused on is the community's complexity of each city. For each sample, we count a number of types on a taxonomy level and compare the difference on community's complexity between cities. And from this boxed plot, we can see there is really the significant difference among the cities' communities' complexity. And we also calculate the KL divergency and find that and discover that the difference between the New York and Sacramento are the largest one. It means the complexity of Sacramento is the biggest one, and for New York is low. The third point we want to find the factors which may be related with the community's complexity, or the phyla or orders or class and so on's abundance distribution. And for this, these figures shows the community distribution of each samples of each city. And from these figures, we can see the difference between samples of the Sacramento is small, and in this city, the Streptophyta, a kind of plant, occupy a large part. And for Boston, the difference between the samples is also big than from the Sacramento, and proteobacteria and the actin bacteria occupy a large part. For New York, the main phyla is just proteobacterial, and the difference between samples is also very big. Due to the data set from the Sacramento provides the station information, so we searched the surrounding environment from the Google map, and we can see the vegetation is rich. So the Streptophyta takes the main part is reasonable. And here I should mention the blue part is identified to the chordata. And to find the relationship between the community's distribution and the factors, we find many phyla's distribution are correlated with the temperature. And for Boston part, this is the result for Boston part. Their main phyla's distribution are also correlated with the temperature, also and the humidity. And I use these figures to show the one of the phyla's distribution, and we can see the phylum named Varrocomi has bias on humidity, and they have the high abundance when the humidity is 65%. And for the complexity part, we find the humidity and the material influence the complexity of the samples in the Boston. And for New York, the phyla's distribution are correlated with the month, and we think month is also related with the humidity and the temperature. And this is conclusion of an analysis published of the MetaSUB. And first, we find the main phyla very different among cities, and the Streptophyta takes a large proportion of Sacramento, and the and the proportion of proteobacterial is higher in Boston and New York. The second one is the difference on communities' complexity between cities is also very huge. And Sacramento has the biggest complexity and New York has the smallest complexity. The third one is the community's distribution is correlated with the humidity and temperature. And for the extension part, I should mention the MetaBinG2 is very useful. And the first one is it requires a small disk space to store. For example, when we do the benchmarking test, we use a 26 and 6 microbials genome and it just requires 72 megabytes. And for clock, this requirement is 24 gigabytes, and for MetaSUB, it's 50 gigabytes. And the update of MetaBinG2 is also very easy because the conversion is independent, and you can transfer the genome's new to the new factors and the older database is okay. And the third one is MetaBinG2 can be scalable to the thousands of data sets, because when we applied MetaBinG2 on this data set with the HPC system, we just used 3 days to complete this analysis. And for the further application part, I want to see the scope of MetaBinG2 is not limited. As I mentioned before, for example, it can be used to do the contamination analysis. For instance, the repeatability is a big challenge of biology experiment, as we all know. And the scientists sometimes don't know where this contamination come from. And with the help of MetaBinG2, it's easy to find this contamination from the laboratory, any corner of laboratory. And I'd like to thank the help of my lab and my supervisor, Dr. Chao Chunwei, and our laboratory members, Zhiqiang Hu, Benjia, and Chen Sun, and I will thank the support of the HPC system of the Shanghai Jiaotong University. And that's all my work. Thank you.
Thanks for the great talk. Any questions from the audience? A couple questions.
Yeah, interesting talks. Especially I like this thing of exploring temperature and humidity. That's a great idea. I was wondering, then you report the complexity, which my understanding is like kind of alpha diversity. How do you deal with the different coverage depth? Or like if you compare Boston versus Sacramento, do you normalize or probably subsample to deal with different number of reads available across the cities?
You mean the samples of these 3 cities are very different? How we deal with these questions? Yes.
Because you do report the numbers of you call it phylum complexity, which my understanding is just the number of different phylum per city.
Yes,
station.
Yes, I know. Because the complexity of these cities are very different, and we can see the complexity of Sacramento is the smallest, and the difference between the stations. The community distribution between stations are small, and for the Boston and for and the New York, this difference are high. But are bigger, yeah. And our MetaBinG2 is designed to just provide the community's distribution, and no matter what kind of samples you have, and for analyze these samples, we just use the traditional statistics method to compare their samples complexity. And you understand, Mamis?
Yep, thank you.
Quick follow-up question. One more in the back.
What is the full scope of MetaBinG2? What all, given the data sets, what all it will do?
Sorry, I can't catch your point.
The MetaBinG2, given some data sets, what are the things that it will do? What is the full scope of this package, MetaBinG2?
The full data set?
For any data set given to Meta, if we want to run MetaBinG2, what all it will do for us? What does it do given a data set?
Oh yeah.
Okay.
It will. Sorry. It will provide a MetaBinG2 is aimed to classify the samples and provide the species levels taxonomy information for each read. And with this result, we can calculate the community distribution downstreams and just provide the information of each read. Yeah, you know.
And you calculate that separately after it's run? Or does
Separately.
Separately. I think that's what there's one other question in the back. Yeah, thank you.
Hi, sorry if I missed this during your talk, but what kind of values do you set for K, both like the order of the Markov model and also the K-mer sizes that you extract from the references? Because, you know, there's kind of a trade-off between the size of your model and its precision. So basically, I'm wondering, like, what values of K did you use?
The K,
Yes.
this K I used is 6, and in the previous work, we have demonstrated this K is feasible to deal with these questions, including the speed and the accuracy.
So is it the same K? So does K represent both the order of the Markov model and the size of the illegal
The size of the
nucleotides?
sequence length.
That's also 6.
Yeah.
Okay, thanks.
Okay, great. Thank you very much. And the last talk before lunch, we'll use LAST plus Megan LR, which is a